This diagram maps out the ultimate ambition of biotechnology. On the left, an isolated ring of microscopic genetic code. On the right, a macroscopic result, a living, transformed organism expressing a new trait. Bridging that gap requires overcoming a severe biological barrier. You cannot simply take a strand of human DNA, drop it next to a bacterial cell, and expect the bacteria to absorb it. Cell membranes are evolved security systems. They act as highly selective barriers designed specifically to reject large, unverified molecules, like free-floating foreign DNA, from entering the internal environment. Recombinant DNA technology bypasses this defense by combining precise molecular tools with living cellular systems, creating a reliable method to smuggle new instructions inside. Acquiring the right genetic code is only the first step. Engineering life requires discovering a stealth mechanism to bypass the host's natural defenses. To achieve this stealth entry, scientists rely on a specific biological vehicle, the plasmid. This diagram of a bacterial cell highlights how plasmids naturally occur. They are small, circular DNA molecules that float independently in the cytoplasm. Their most critical feature is their autonomy. Plasmids replicate on their own cycle, completely separate from the main, tangled bacterial chromosome. Researchers exploit this independent replication. Because plasmids are small and modular, they can be co-opted as vectors, delivery vehicles that carry foreign genetic material directly into a host. By splicing a target gene into an autonomous vector, scientists create a biological Trojan horse. It enters the cell and multiplies the new gene, without triggering the host's primary replication limits. Before the plasmid can carry anything, researchers must open the ring and insert the new code. This requires restriction enzymes, which act as highly specific molecular scissions to sever the DNA backbone at precise chemical locations. Looking at this schematic, both the plasmid DNA and the foreign gene must be cut using the exact same restriction enzyme. This leaves short, single-stranded overhangs of unpaired nucleotide bases, known as sticky ends. Because the cuts are identical, these sticky ends are perfectly complementary. Through standard base pairing rules, hydrogen bonds naturally pull the foreign gene fragment into alignment with the open plasmid. The bases pair up, but there is still a structural vulnerability. The rigid sugar-phosphate backbone of the DNA molecule remains physically broken at the splice points. Chemical complementarity aligns the foreign DNA, but a physical mechanism must step in to permanently lock the structure in place. To fix the broken backbone, the assembly line requires a molecular welder, the enzyme DNA ligase. This animation illustrates how DNA ligase specifically targets the fragile junctions where the foreign gene fragment meets the plasmid vector. As it moves across the junction, the ligase forms new chemical bonds, permanently sealing the sugar-phosphate backbone into a continuous structure. The result is a fully intact recombinant plasmid, a hybrid molecule containing instructions from two different species. However, a perfectly assembled recombinant plasmid cannot execute its code while sitting in a test tube. Splicing writes the instruction manual. Now that manual must be forced into a biological machine. This brings us to transformation, the active process of introducing foreign DNA into a living cell. As established before, bacterial cell membranes are inherently hostile to large DNA molecules trying to cross into their internal environment. These diagrams illustrate two common techniques used to bypass this defense: electroporation and heat shock. By subjecting the bacteria to rapid temperature shifts or electrical pulses, scientists temporarily increase the permeability of the cell membrane, creating microscopic physical pores. While the membrane is destabilized, the recombinant plasmids suspended in the surrounding fluid slip through these pores and enter the interior of the cell. To be clear, transformation is a biochemical term. The cell does not mutate its physical shape, change species, or visibly alter its outward appearance. As this diagram demonstrates, the newly introduced plasmid does not replace the main bacterial chromosome. It coexists alongside the host's original DNA. When the shock treatment ends, the cell membrane quickly reseals, permanently trapping the vector inside. It is a highly inefficient, low-probability event. Scientists must carefully expose millions of bacterial cells to these laboratory conditions, relying on the statistical chance that a small fraction will survive and successfully accept the vector. Overcoming the physical barrier of the membrane is a violent mechanical necessity. It is the only way to establish the host-vector relationship. Once the membrane reseals, the surviving bacterium recovers and resumes its normal biological functions, which includes the physical process of cellular division. This diagram shows the immediate consequence. Because the plasmid replicates independently, both daughter cells inherit the recombinant vector when the cell splits. This triggers exponential amplification. Multiplying into a massive colony, the bacteria produce thousands of identical copies of the inserted target gene. But possessing the multiplied code is not the final step. A cell can carry a foreign gene without ever actually using it. The bacteria have been hijacked into serving as self-replicating copying machines. Yet, merely copying the code is vastly different from reading it and building the protein it describes. For the inserted gene to function, it must be paired with specific regulatory sequences. The host cell needs a chemical signal indicating that baflorin DNA is a valid instruction worth reading. If those sequences are present, the cell begins gene expression, the active process of transcribing the foreign code into messenger RNA and translating it into a physical product. This cross-section of gene expression shows the host cell's ribosome blindly reading the foreign RNA. It processes the instructions and begins assembling the encoded protein exactly as if it were its own. This diagram illustrates the final output of this process at scale. By demanding vats of transformed bacteria to run this exact translation cycle, the pharmaceutical industry manufactures massive, purified quantities of critical human proteins, like insulin. The entire process relies on an unbroken biophysical chain, from precise restriction enzyme cuts to forced membrane shock, and finally to hijacked ribosomal translation. Recombinant DNA technology succeeds by mastering the rules of molecular chemistry. By leveraging those rules, biological systems are forced to execute foreign code, turning simple cells into programmable microscopic factories.